to make sure that that little bit gets in there, but I will cover it today. So in case you're curious about intermediate filaments, microtubules, or actin filaments, that's coming in. We'll have that today. All right, we'll see how this goes. Great. All right, any other questions? All right, well then we'll get started. Some of this will be review for you, if you remembered anything at all from last semester. But we have to do this. We have to review our neurons and our glia, because what if you forgot? I don't know about you, but I forget things from time to time. So we're going to make sure we're all on the same page before we forge ahead. Some of this will be new, and then some of it will be review. And when you know it already, pat yourself on the back. Great job. We're going to talk about the morphology of neurons. A bit about why that matters, because it does. There's a reason why certain neurons are shaped a certain way. What creates that morphology? We'll talk about synapses, of course. The purpose of neurons is to communicate, and synapses are the site of communication. There's two broad categories, more on that later. And then we'll go through the glia. They get that name because they were thought of as just the glue that held the brain together. They weren't thought of as functional units, they were just packing peanuts. Of course, that's not true. And we'll go through that and give them the attention that they deserve. Neurons are highly specialized cells, and they're specialized for communication. That's their job. Talk to each other, talk to a target tissue. So they have intricate morphologies that allow them to carry out this special function. When we think of neurons, we think of them as polar. And there's a couple meanings to that word. The polarity across the membrane is going to be the focus of lectures 2, 3, and then probably all the rest of them too, when you get down to it, because that's how neurons work. By adjusting their membrane polarity, by changing the membrane potential, that's what determines their function. So there's that polarity. But the polarity I'm talking about on this slide has to do with the two ends of a neuron, the receptive end and the transmissive end. Communication requires both. If you've ever tried to have a conversation with someone who doesn't listen, it doesn't work very well. And the same thing's true if they never respond. So there has to be a back and forth. There has to be an input and an output in order for communication to work properly. And neurons have both of those. Those ends would be the dendrites and the axon. Every neuron, of course, has a cell body. We're probably not going to talk a ton about the cell body. It's great, though. This is the cell. If you've ever taken a biology class and you've learned about the cell, here it is. This has all those organelles you learned about. Everything that a cell needs to do goes on in the cell body. Mainly it's gene expression. If something happens in life, a protein did it. That protein had to be made, and that's going to happen in the cell body. But when we're thinking about polarity, we're thinking about the dendrites and axons. These are what make neurons special. Dendrites are variable. So are axons, but dendrites are a little more variable. There can be anywhere between 0 and a bunch that come off of the cell body. And this is what's going to give the neurons these different names. Unipolar, bipolar, multipolar. Those poles are the extensions that come off. If they're unipolar, that means there's only one. It has to be the axon. A neuron's not much of a neuron without an axon. So unipolar cells don't really have dendrites. Here's a unipolar cell. Here's the cell body. Here's the axon coming off. Then there's a little specialized extension here that acts as dendrites. They're not really dendrites. We don't have these, but we do have these pseudo-unipolar cells. We call them pseudo-unipolar because they start off as bipolar. Early on in development, they have two poles. They got their axon, and they got one dendrite. 1 plus 1 is 2. That's about as complicated as we're going to get with the math today. But then throughout development, their cell body shifts over, they fuse their axon and dendrites, and that's what creates the pseudo-unipolar cells. In case you don't care about these yet, these are the cells that allow you to feel. So you should care about these. Your dorsal root ganglia are made exclusively of pseudo-unipolar cells. At least the projection neurons are all pseudo-unipolar. They don't really have dendrites. They have little specializations of the axon that are able to receive input from specialized receptors. They don't really have dendrites. In the central nervous system, when you're within the brain, you're going to see a lot of multipolar cells in the brain and spinal cord. Multipolar just means they have a lot of projections coming off of them. They have more than two, that's it. That's all it takes to be multipolar. But because they have additional dendrites, they have additional sites of input. That's what we're going to think of dendrites as. And that's pretty well true, except when it's not. But we should think of dendrites that way. They are the ears of the neuron. It's the receptive end. When you have multiple dendrites, you have multiple sites of input. You can listen to a greater number of sources, so you can integrate information. That's why we see these in the central nervous system. As we went over yesterday, the central nervous system is where we integrate information. If you're going to integrate information, you need to take multiple bits into account. Bipolar cells and pseudo-unipolar cells, we see a lot more of these in the peripheral nervous system, where we're simply transmitting from A to B, kind of a linear fashion, from my retina to my optic nerve on back to the thalamus. We don't need to make sense of anything, just get it in there and let these make sense of it. So there's a reason why neurons have different numbers of dendrites. If you need to do something really complicated like coordinate movement, you need the dendritic arbor of a Purkinje cell. That's what this is. Very elaborate dendritic trees, and that allows them to have hundreds of thousands of synapses. So you can modify your movements based on experience. The dendrites may or may not have these little projections called dendritic spines. These are fairly important for learning because they are volatile sites. They emerge, they disappear based on experience, so you can learn and forget, respectively. Not every neuron has these. Some are a-spiny, and then others are spiny. You see these little protrusions coming off? Here's your dendritic shaft. Here's all the little spines. They come in different shapes and sizes. The bigger the spine, the stronger the synapse. So when you're creating a memory, you're likely increasing the size and number of dendritic spines in that particular network that encodes whatever we're talking about. Maybe it's the fact that elevated intracellular calcium levels lead to via mitochondrial permeabilization. So we'll get there. You got some spines hanging around, but if you don't use it, they're going to go away. These spines are created and lost, and that's what allows us to be flexible with what we know. So they're kind of important. Also important would be the axon. There's always one of these. There's never two. There's always one. I'm not going to say except when there's not. There's always one. Now that one axon can branch extensively, so here's our little neuron here, and here's their tracing of the axon, and when it reaches the place that it wants to innervate, this is still one axon, it just branches a bunch, so it can contact multiple neurons. One axon. This is the mouth. This is how we're going to communicate to the target cell, whether that be another neuron, a muscle, a gland, doesn't matter. Every neuron is going to have these general segments. Input, usually that's the dendrites, could be the cell body, could be the axon, you never know. Some site of integration, that's usually going to be around the cell body near the initial segment of the axon. Then some conductive portion, that's always the axon. Well, I'll go ahead and say that's true. And then the output, which we'll think of as the axon as well. This is the polarity. We have sides. There are two sides, input and an output. A little bit happens in the middle, and that's so we can process information. But regardless of the neuron, they all have this. Whether they're a fake neuron on a computer, pseudo-unipolar, multipolar, whether they're communicating with muscles, neurons near and afar, or blood vessels, it doesn't matter. All neurons have to be able to receive input and generate some output. We should be thinking of those as taking place in the dendrites and axons, respectively. Those elaborate morphologies that we see, all those dendrites, the axon, the cell body, everything is shaped by the cytoskeleton, which just means cell skeleton. And there's 3 filaments that we find in every type of eukaryotic cell anyway. The intermediate filaments we call neurofilaments because it sounds cooler. These are not used for anything other than structure. That's it. These are the rebar of the cell. They provide strong mechanical support. That's about all we're going to say about them. We'll talk a bit more about microtubules and microfilaments in Lecture 7 when we cover axonal transport, because these 2 filaments have polarity. There's a plus end and a minus end because they're made of 2 different subunits. So transport proteins like kinesin and dynein can tell which way they're heading. Am I moving away from the cell body and down the axon? That's what we call anterograde transport. Or am I heading back? So here we can see the distribution of these different filaments. So these microtubules, which are shown in yellow here, they radiate out from the center. So we know where we're headed. And there's a reason we have to head down the axon. Why is that? What's that?
It was this.
What was it?
Myelin.
Myelin?
For conduction and go through the nerves. It's wrong. Moving on.
How the ion concentration works?
I like all the words that are coming up here. We'll get to that in just a bit. We'll revisit this. The microfilaments shown in purple here are mostly concentrated in the periphery. This is what gives the cell its fine shape. So these are going to be found in your dendritic spines. The microtubules, very important for long distance transport. This is critical for the axon because we don't make protein there. And proteins do everything for us. So if you'd like your axon to function, you're going to have to ship everything down. And it's a wonderful thing that our microtubules are polar. They orient their plus end toward the axon terminal. So kinesin knows I'm heading the right way, and dynein can take junk back for recycling. So we can refresh and recycle. That's the focus of Lecture 7, so we will revisit that again. The microfilaments, actin filaments, these are what's going to give the cell its actual shape at the edge. Microtubules provide the roads, and then the actin filaments are going to create these little extensions like the dendritic spine here. They're going to help shape the presynaptic site as well. Here we can see actin in an actual dendrite. So green is just showing you the cell shape, and then they've stained actin red. Red plus green equals yellow when we're dealing with light. So here's actual dendritic spines. Notice they're not all the same size and shape. The bigger ones have stronger synapses, and these long, thin ones, weaker synapses, probably those that are going to wither away. And here's the proof that we must transport down the axon. This is a stain for Nissl substance. We should remember this. It's back and it's still true. Nissl substance would just be all that stuff that you need to create proteins. The cell body houses the nucleus. That's where we're going to make RNA. It's also filled with ribosomes, rough ER, and Golgi. So we can turn that RNA into protein and shuttle it to the correct location. You'll find some Nissl substance in the dendrites. Here's some dendrites. You'll see they're very lightly colored in purple. And then fading into the background is the axon. It's devoid of Nissl substance. So all the proteins that are in presynaptic sites were first made here and then transported along those microtubules. When this doesn't work properly, synapses wither, neurons die. See last semester. Another important aspect of the soma is the fact that dendrites emerge from it. In those multipolar neurons that are trying to take into account multiple pieces of information, that site would be the cell body, because all dendrites emerge from it. So all those electrical events that are being created in dendrites, in this case 1, 2, 3, and 4, they all reach the cell body where they can sum together. This is where that electrical information gets integrated, and the neuron makes a decision. Should I tell my neighbors or not? If it does, It's going to conduct an action potential down the axon and tell everyone about it. So not only are we making proteins so that we can do everything, we're also integrating input from all of our neighbors so we can decide whether or not we communicate to our target. So the cell body is fairly important. Also important would be the axon. This is where we're going to conduct electrical signals called action potentials away from the cell body and toward our target, whether that be another neuron, a muscle, a gland. An important part of the axon is the axon pillar. This is that initial segment of the axon just coming off of the cell body. Here's the cell body of a neuron. They've stained it with a dye called sulfurrhodamine, and then they labeled sodium channels with an antibody. So they're showing up green here. Red is just showing you the neuron's morphology, and green is showing you sodium channels. Here's the axon. Notice we don't have any green up here. The cell body is not going to generate these action potentials. It's the axon. There's a couple really good reasons why it can do this. On this slide, we can see all the sodium channels there. We can also see that it's very, very thin compared to the size of the cell body. So it has much higher resistance. More on that in lectures 2 and 3. No, lectures 3 and 4. Sorry. Lectures 3 and 4 are all about that. Now the axon can be myelinated to speed up signal transduction, so the action potential moves more rapidly. We'll talk about why that's the case in lecture 4. But whether you're myelinated or not, you have to have these sodium channels. They are what create the action potential because they allow sodium to rush in. You can actually measure that here over time. They're showing you color-coded the intracellular sodium concentration, and here we can see a spike of sodium right at the initial segment. You won't see this in the myelinated segments. We don't move ions across the membrane there. But here is the beginning of the action potential, and there it is. This is what lecture 4 is all about. And this is what neurons use to rapidly conduct signals within themselves. The pattern that we see is electrical, chemical, back and forth all day. Electrical within neurons and then chemical between. Here's that electrical event that stimulates chemical release. When neurons aren't excited sufficiently, only minor changes in membrane potential. That's what we're looking at here. So this is time on the X, and here is the membrane potential. Usually we rest negative, around negative 70, 62 is close enough. During an action potential, we actually cross zero, we have a positive membrane potential, and that's what stimulates neurotransmitter release, and then we come back down. It's going to be the most exciting 4 to 5 milliseconds of your life when we talk about that. This is going to travel down the axon. It's going to propagate itself, kind of like fire. We'll talk about why that's the case. And that's going to stimulate neurotransmitter release. There's that chemical. Electrical, chemical, electrical, chemical. That's the pattern. The purpose of chemical release is to elicit some sort of change in the target cell, whether it's another neuron or something else. And usually this is going to be a change in membrane potential. Neurons, muscles, same thing. We're going to hopefully elicit another active potential and keep everything moving. Do we have any questions?
Nope.
Thank you. I don't think I do either.
Yeah, you do.
Oh, yeah, you're right. Okay, take a moment to talk about these, and we'll go through it. Well, then take it away for us and tell us about the soma, dendrites, and axon, what they do for neurons.
The soma is the cell body, makes protons, dendrites.
Hold on now. Protons?
Proteins.
Thank you. It does also make protons.
Dendrites receive info, and then axons like send out info.
Excellent. Nice. Speaking of dendrites, multipolar, bipolar, and unipolar. Rita.
Multipolar have one axon and multiple primary dendrites. Bipolar, one axon and one dendrite, and then unipolar is a single one that acts as both axon and dendrite.
Fantastic. Which one of these do you think you're more likely to find in the central nervous system? And why? Why?
Because you have a lot of connections and stuff.
There we go. Yeah. We can integrate additional information. We're not just passing along. Here you go. I saw this much light. You figure it out. All right, so we have 3 different types of cytoskeletal filaments. Tell me what they are and what they do for us. Leah. You're welcome.
Okay, so we have neurofilaments, and some are those just for support.
Okay.
And then microtubules, that is in the axon and it allows like the proteins to be transported electrical signals. And then microfilaments are near the membranes and synapses, and those allow for local trafficking in cell shape.
Okay, I like that for the most part. I think that's really good. Let's just keep in mind that the microtubules are everywhere. They radiate out from the center of the cell. You'll find them in the dendrites, you'll find them in the soma, you'll find them in the axon, but they are really, really important for trafficking because they don't move around that much. Actin filaments move around a lot in the periphery. Intermediate filaments are also throughout. Anywhere you have a cell, you're going to have all 3 of these. But we've organized the microtubules in the cell body in the center so we know direction. I won't ask why. Okay, let's hear about action potentials very briefly because we will hear about them in great detail in lecture 4. Stephanie Smith.
Action potentials are a result when you have enough input that stimulates the action potential that pushes the electric signal that goes down the axon to result in a chemical release from synapses.
Fantastic. It's all about communication. So its function is to stimulate chemical release. And it's just some kind of electrical signal. It's going to last a couple milliseconds. Sometimes it lasts only a few hundred microseconds. It's variable. But it's a very brief electrical signal used to elicit chemical release. Is there anything you'd like to go over before we move ahead? Okay, then we'll move ahead. Neurons are only there to communicate. The only reason that they generate action potentials is so they can spit out chemical transmitters and affect the activity of some target cell. The area where this happens we'll call the synapse. This is that junction between the neuron and whatever it is that it's talking to. In the central nervous system, 2 neurons are going to be on both sides of the synapse. Out there in the periphery, it's probably a neuron and a muscle. There are 2 types of synapses. There's electrical and chemical. That's nice. Because those are the 2 methods of communication that neurons have. Electrical within, chemical between. Except when it's not. Sometimes it's electrical between. This is rare though. This is rare because it's not very flexible. And what it does is force neurons to work as a single unit. So it limits the processing power because they have fewer unique patterns that they can create. But it amplifies the response. Do we have any photography enthusiasts here? Perfect. So you can reduce the resolution of your image in low light settings to increase the gain. So you ever take a picture and it's dark and it's all fuzzy? You ever take a selfie, maybe it's nighttime, and it's a little fuzzy, it's grainy. It's not as crisp as if you were to go outside in public and take that selfie, but you don't want to do that. But when it's bright, we don't have to bin our pixels together, so we get full resolution. When it's dim, we have to bin pixels so that we can add up the light in all 4 of these so that we can actually approach a visible level of light. That's kind of what's going on here, and I'm glad I just confused everyone here with that. But the way that neurons bend themselves together is through electric synapses, so that they can all work together. So rather than just one neuron spitting out neurotransmitter, now you have 100, but they all act together. So you don't get to encode any other patterns. They're all on or they're all off. The way that they act together is by creating these continuous pores where they actually share their cytoplasm. So the electricity in one cell can directly transfer to the other, and that's what we call gap junctions. They're created by these connexin proteins. Here's a cartoon version, here's an electron micrograph showing you these little tiny pores. All of those little donuts there are connexin proteins. Yeah?
So it sounds like you're saying they continuously make them. Is a gap junction not something that's always there between them?
They make it and they keep it there, yeah.
So once it's made, it's always there?
Correct, yes. So when they fuse themselves together, now they're a single computational unit, essentially. So they're just going to work as one for the rest of their lives. They're committed. They share their cytoplasm. There's a continuous pore between cell A and cell B, so that whenever you have a change in membrane potential in one cell, you get a corresponding change in the other. That's what we're seeing here, believe it or not. Let's walk through it. We're looking at paired recordings of fast spiking neurons. These are local inhibitory neurons, and pyramidal neurons. These are excitatory projection neurons, just in case you care. Doesn't matter for the purpose of this slide. These two neurons happen to share electrical synapses. They have gap junctions connecting their cytoplasm. So what they've done here, here's 1, 2 pipettes. They are attached to both of these neurons, and what they'll do is deliver an electrical current, that's this here, to one cell. And then they'll measure the membrane potential in both. When you have gap junctions, and you depolarize one cell, the other one depolarizes as well. They're acting together. It's not as much depolarization because gap junctions have resistance, but still, they're acting together. Depolarize one, depolarize the other. Hyperpolarize one, hyperpolarize the other. Yeah.
So I'm assuming like the physics still apply. Is it a predictable amount of loss between the voltage when you go from gap to gap?
Oh, it totally depends on the gap junction. Right. How many of those connexin proteins are there? So that's what's going to determine the resistance. And V equals IR. That's always true. Yeah. So you can tell how well they're coupled by looking at this. Yeah, exactly. Now, these two neurons, no gap junctions, no electrical synapses between them. So they're discrete computational units. You can depolarize cell 1 all you want. This cell doesn't care. And this allows them to act independently so you can create unique patterns. They're the output. We want them to act independently. But these local neurons are just going to shape activity. We want them to act together. These are rare, though. We probably won't talk about gap junctions too much. I think I mentioned them in one more lecture, but I've rearranged so I don't have the number up here. Sorry. For the most part, what you're going to see are chemical synapses. These are a lot more flexible. One neuron that's excited can inhibit the other. We can have a change in polarity. It's not always moving in the same direction. We can also create long-lived changes as opposed to just the immediate brief change in membrane potential that we get at the gap junction. So this is the type of synapse you should be thinking of when you think synapse. Are you thinking it? Okay, good. And this is what you should see. And you should be able to pick out the presynaptic side, not because they've written it on here, but because there are these synaptic vesicles. See these little balls here? Those are little packets of neurotransmitter. Those chemical signals that we're going to release into the synaptic cleft. That's this little 20 nanometer gap here, really tiny space. Here's where the chemical happens, ladies and gentlemen. Electrical goes to chemical just right here, and then it's back to electrical. It's very brief, but very, very important. The postsynaptic side of things, notice it's devoid of those synaptic vesicles. This is the receptive end. Which part of the neuron might this be? Dendrites, yeah, exactly. Look for the synaptic vesicles. These are going to fuse with the membrane and release neurotransmitters. More on that in lecture 5. The postsynaptic side of things are going to translate that chemical transmitter into an electrical event. More on that in lecture 6. Notice how electron dense it is. That's just telling you there's a ton of proteins here. There's a lot going on with these synapses, despite being so small. These are the overwhelming majority of synapses that we have. So just think of this. Realize that electrical synapses exist, but they're rare. They are exceptions. Chemical synapses are very flexible. Typically, we're going to be thinking of axodendritic synapses or axosomatic, but you should be aware that chemical synapses can form between any of those elements in neurons. You can actually have somatosomatic synapses where the two cell bodies form a synapse. Typically, though, here's what we're dealing with. Axodendritic, which can be excitatory or inhibitory. Now, what that means, excitatory synapses would be those that increase the likelihood that the target cell is going to fire an action potential. That's it. They depolarize the neuron and bring it closer to threshold. That'll make a lot more sense after lecture 4. Inhibitory synapses are those that either shunt that excitation or that hyperpolarize the target neuron. They make it less likely that it fires an action potential and communicates with its target. So they're exactly what they sound like. An excitatory synapse excites that postsynaptic neuron, and an inhibitory synapse inhibits it. And we're talking about exciting and inhibition. We're talking about action potentials. Are they going to generate an action potential because of excitatory input, or are they not because of inhibitory input? Typically, your excitatory synapses are going to be more distal in the dendrites. They're further away because the closer synapses tend to be inhibitory. This allows the inhibitory input to essentially head off any excitation out here, so we can trump that excitation. So you tend to find inhibitory synapses on the proximal dendrites and on the cell body, so that inhibition can cancel out that excitation. We always want to bias toward inactivity. This way we're not wasting ATP. Really important. If you see a synapse on the spine, that's always excitatory. But there's variability here. It's not always axon, dendrite, axon, cell body. You can have from axon to another axon. And these can be excitatory or inhibitory. It depends on what kind it is. These are going to act either on presynaptic sites or at the initial segment to stop an action potential. At presynaptic sites, what they do there is fine-tune the amount of neurotransmitter that's released. They can turn it down a bit by inhibiting that calcium current that comes in. So here we're looking at the amount of calcium that comes in. This is normal. And here's what that inhibition, less calcium translates to fewer neurotransmitters released. And so the postsynaptic potential here, so this is recorded from cell B. There's their fate. But we don't get the same degree of excitation because we've decreased calcium input here. That'll make a whole lot more sense after lecture 5. Or we can facilitate. We could provide excitatory input right here where it matters so that we spit out a little bit more neurotransmitter, or perhaps even a different neurotransmitter. And then, of course, the dendrites can form synapses as well. These are rare. They exist. Just know that they exist and don't think about them much more. Now, chemical synapses don't have to just have that 20 nanometer gap. Sometimes they operate by what we call volume transmission. This is going to be true for your neuromodulatory transmitters like your monoamines. Dopamine, for example, can operate by volume transmission. Glutamate does not. If glutamate operates by volume transmission, neurons die. I hope we remember that extrasynaptic glutamate receptors are toxic. I hope so. We don't want that glutamate to diffuse away, and we'll see what happens when we let glutamate diffuse away later in this lecture. We also saw it last semester, but you never know, you might forget. So in volume transmission, what we're doing is releasing neurotransmitters not right next to the postsynaptic site. So here's your typical chemical synapse. We release it further away and allow it to diffuse to multiple sites. So this allows the dopamine in this example to diffuse away, and rather than targeting a single postsynaptic site, it hits multiple. Sure, it's a lower concentration, but it hangs around a little bit longer, and you contact additional sites. So this allows you to get a more uniform response in that nucleus that you're communicating with. This is especially important for dopamine because it tends to act over a large area and over a long time. This sort of non-traditional release is also true for peptide transmitters. Most neurotransmitters are very small. They're derived from a single amino acid. More on that in lecture 5, I think. But they're small. They're not multiple amino acids linked together. That's the peptide transmitters. These are non-renewable. We can't recycle these. They're expensive. We have to make them in the cell body and transport them all the way down to the end. That takes time, it takes energy. So we don't want to just release them willy-nilly. So we hold these dense core vesicles, what we call them, away from the synapse. As we will see, when the action potential travels down into the presynaptic site, it's going to cause local calcium influx to stimulate neurotransmitter release. Local is the key word there. But as the neuron fires action potential after action potential in a burst, that calcium builds up and will eventually reach the extrasynaptic sites and allow for peptide release. So this allows a neuron that's really, really active to send a completely different message to its target. These neuropeptides could be excitatory, while the small molecule that it's releasing might be inhibitory. So that when it's normally active, just a little bit, it inhibits, but when it's really active, then it excites. There's a lot of flexibility to chemical synapses. That's the message today. The specifics of neurotransmitters and their receptors will come up in lectures 5 and 6, and you'll hear all about lecture 6 repeatedly throughout this class, so please give that one a solid read. If GQ, GS, and GI don't make sense to you right now, boy, they will at the end of this class. Because that's how everything works. Do we have any questions? No? Cool. Take a moment, talk about these, and then we'll work through them. Okay. How are we communicating at an electrical synapse, and why do we have them? Jess?
Hi.
Hi. Tell me about electrical synapses.
So they're pretty rare and not very flexible because it's when they all connect together to make the gap junctions. They work as one unit to amplify a response so that it mimics the pattern.
Great. So, let's say that I have two cells connected by a gap junction. One of them depolarizes. In other words, its membrane potential becomes more positive. Does the other one depolarize as well?
Yes.
Yeah. Is there ever a case, though, where it might hyperpolarize while this one is depolarizing?
She's nodding
Probably not. Probably not. I mean, there might be some rare things going on, but the point is they act as a
We're
unit.
all as one.
Yes. They're not independent processors at this point. So they're a little less flexible, but they're much louder. That's the point. You get a more robust response, but it's the same response every time. Okay. Chemical synapses have a few parts. You can think of it as two. A lot of times we call it three. We'll get to that third one later. I just want to hear about at least two, Callie.
So there's the presynaptic area, and that brings the electrical signal and turns it into a chemical signal. So it releases the neurotransmitters into the synaptic cleft, where they diffuse across the membranes, and enter the postsynaptic area, which takes that chemical and turns it back into an electrical signal.
Fantastic. You got really two parts, if you don't want to count the presynaptic, postsynaptic. Don't overcomplicate it. Presynaptic releases neurotransmitter, postsynaptic receives and converts that to an electrical event. Okay, excitatory or inhibitory. I'm going to be using these words. It would be nice if we all knew what they meant. Sydney?
Is the other one
No.
Okay.
It's Neil.
Okay. Excitatory, it increases the likelihood that an action potential will be generated, and inhibitory decreases the likelihood that an action potential will be generated.
Fantastic. That's it. We're going to depolarize, or we're going to not. We're going to prevent depolarization or cause hyperpolarization. Very good. All right, we got a few different types of local chemical synapses. Which of these are the most common, Stephanie?
The axons and the axons.
Great, great. And so the order here is just telling you what's presynaptic and what's postsynaptic. Go from axon to cell body, axon again, right? Excitatory or inhibitory? Yeah, those are usually going to be inhibitory. We want to bias toward inaction. This is going to come up throughout the class, especially when we talk about the basal ganglia. It's better to not do. It's going to waste energy if it's not the right thing to do. How about axodendritic? Excitatory, inhibitory? It could be either one. Do we have some kind of distribution of excitatory and inhibitory based on location?
So more proximal is going to be more inhibitory and farther away is going to be excitatory.
Fantastic, right? These are inhibitory. The ones in the dendrites that are close to them are also inhibitory. Again, so we can bias toward inaction. How about axonic?
Typically inhibitory.
Okay. Are they always? No. Yeah. It depends. So right there at the initial segment of the axon, those are usually inhibitory at the presynaptic site. It could be either one. It depends. And same thing for dendrodendritic. It depends. So for pretty much all of these, it could be either one. I'll just have to tell you. Yeah, that makes it nice. Now, not every synapse just has that 20 nanometer synaptic cleft. That's why I didn't want to count it. Sometimes we operate by volume transmission. What the heck is that? Mackenzie.
So volume transmission involves neurotransmitters being released from presynaptic sites that aren't like right near their postsynaptic site. So instead the neurotransmitters go through a slower diffusion process to reach multiple postsynaptic sites.
Fantastic. There you go. Yep. So we can hit multiple parts. All right. We'll talk about glia. We'll call that lecture 1. Then we'll be introducing new things. There's a few different types of glia, and they're all going to do different things to keep neurons operating. I like to think of neurons as little children or little babies, and so they cry a lot. That's all they do. They cry and they make messes. The glia are the mother or the father. We'll just say parent. Maybe they're a stay-at-home dad. I don't know. I was. They're there to clean up the mess, to minimize the mess. They're there to support the neurons. Without them, neurons would kill themselves. This is also true for children. We've seen this before. The ependymal cells are the glial cells that create CSF. Does this look familiar? Fantastic. If you were awake yesterday, you should remember this. So here we can see our ventricles. What are those ones off to the side? Not at the midline? Yeah, there you go. All throughout our ventricles, we have a lining of ependymal cells. That's what we're seeing here on the left. Here's the ventricle. This should be filled with fluid, and then here we can actually see parenchyma. So this is cortical tissue here. And lining that, if we zoom in here, that's all this is. Here's the ventricle. Now they've just turned it a bit. And here's your ependymal cells lining that. They have these little projections that are going to kind of float and keep the CSF moving. And they're going to create CSF. They create it all throughout your ventricles, but primarily they create it at the choroid plexus, shown here as little red balls. And they're red because of all the blood vessels there. They create a greater volume of CSF here because they have better access to blood. That's where they're pulling the fluid from anyway. So at the choroid plexus, we're going to create a greater volume of CSF, but we're making it all throughout. Just keep that in mind. Ependymal cells make and move CSF. That's their job. The myelinating glia come in at least two types. There's the oligodendrocyte and the Schwann cells. There are different classes of oligodendrocytes, not all of them myelinated, it turns out. And the same thing is true for Schwann cells. There's also non-myelinating Schwann cells. But when we think oligodendrocyte and Schwann cell, we want to think myelin. And that's true except when it's not. In this case, it's true. And what myelin is this lipid-rich membrane that gets wrapped around the axon. So here's our cartoon neuron here. Would this be unipolar, bipolar, or multipolar? Multipolar, yeah, say it loud. Say it proud. Here's our axon initial segment. All these little balls would be sodium channels so we can create our action potential. And here we have the myelin sheath. This looks a whole lot like a peripheral neuron to me because those have to be Schwann cells. Schwann cells only myelinate one segment, whereas oligodendrocytes, because they're in the central nervous system, will myelinate multiple segments to save space. And they also have very compact myelin. Here's a cross-section through an axon. You can see the myelin sheath, layer upon layer of fat. Its job is to insulate the axon. As we will discuss in Lecture 4, it's going to facilitate charge transfer down the axon. And it does that by reducing membrane capacitance. That's its major function. It also dramatically increases membrane resistance, and that helps too, but it doesn't speed it up quite as much as the capacitance. Apparently I've moved on to microglia. Despite their name, they're not actually nerve cells. They are derived from the yolk sac early on in development. So these are little blood cells that make it into the nervous system before it's closed itself off from the outside world. We've heard of microglia before. Here they are again. They do the same thing they did last semester. They are going to be the resident macrophages of the nervous system. They're there to clean up in case there's tissue damage or any foreign invader who might want to take root in your nervous system. Their job is to go and consume that. Now, normally they look a whole lot like a basic nerve cell. They have their cell body and they have these little projections. They put out these little extensions. They look kind of like dendrites. They're not, but they look kind of like them. In response to injury, they round up, look a little bit more like an amoeba, and they act as macrophages. They'll go find whatever it is that seems foreign and eat it. That's their job. They, of course, play a major role in detecting and prolonging inflammation. That's still true. None of that has changed. When we think microglia, we should be thinking macrophage, injury. They do way more than that, but we won't talk about it. The last glial cell we're going to talk about today would be astrocytes. It means star cell because they kind of look like stars, I suppose. Here's a bunch of astrocytes shown in white. The way that they show you astrocytes is with this glial fibrillary acidic protein. That's an intermediate filament. Notice how it's scattered all throughout the cell, sort of. Their morphology is a lot more intricate than this, but we're not doing an actin stain, so we don't see the edge. But here we can see astrocytes closely associating to this thing right here. What is this? Any idea? That would be a massive axon. That's a blood vessel, turns out. But it's a good guess, because they do associate with axons. But I'm talking about their input processes here. That's the extension that they put out and contact blood vessels. The purpose of that is to pull out water, ions, glucose, things that we might want from the blood, to pull it away and more directly transmit it to brain tissue. We've heard of the glial limitans. Yesterday, maybe? Excellent. Bunch of input processes. They're there to make sure nothing nasty comes into the nervous system. Now, there are other processes that astrocytes extend because they have many jobs. Not only do they help preserve the integrity of the blood-brain barrier through these input processes and help bring in nutrients and remove waste, they also help clean up the mess that neurons make when they conduct action potentials down the axon. Again, neurons are little children, and when they play, they make a mess. And so when they propagate an action potential down the axon, they spit out a bunch of potassium, and that needs to be cleaned up. If we have an elevation of extracellular potassium, well, we might run the risk of having a seizure. We don't want that. We talked about this last semester. Those aren't good. So to prevent that buildup of potassium, we have perinodal processes. They contact those nodes between each myelin sheath. And when the potassium comes out, astrocytes clean it up. so we don't build up our extracellular potassium levels. They stay nice and low, and this way potassium stays inhibitory rather than excitatory. And that'll make a whole lot more sense after lecture 3. And then we have carrysynaptic processes. Just like it sounds, they are near synapses. Here's our pre- and postsynaptic site. How can we tell the presynaptic site? The vesicles, the word, exactly. This is the cartoon version of that electron micrograph that we saw earlier on. It's the same image, they've just cleaned it up a little bit so it's easier to see. Now the reason that I said sometimes people call synapses as having 3 parts is because there's usually a glial element there as well to help clean things up. Children make messes. And that's a part of their job. That's what my mother tells me anyway. When they spit out that transmitter, it has to be cleaned up. Otherwise, it's going to keep binding to its receptor. And it's just as important to terminate a signal as it is to create it in the first place. You need on and off. If there's no off, it's just noise. So, to make sure that our chemical transmitters don't become noise, we have these carrysynaptic processes that have glutamate transporters, for example. Proteins that transport neurotransmitters inside the astrocyte so that those levels don't build up here. If we have glutamate build up, neurons die. If we have potassium build up, neurons die. So again, we're taking care of the children here. These are the mother that's going to clean up the mess, whether that mess be an ion or a neurotransmitter. We got to clean it up so they can continue to play with each other. The most important functions we've already gone over: cleaning up the ions and neurotransmitters, picking things out of the blood, glucose, shuttling pyruvate over to neurons. We won't talk much about this, but neurons are primed more for aerobic metabolism. So they're going to be using primarily pyruvate to generate their ATP. There's not as much anaerobic metabolism, so they're not going through glycolysis as much as the glia are. Glycolysis has a low payout anyway. So neurons are using a lot more ATP. They care more about the aerobic side of things. So they can avoid some of those toxic byproducts that you create in anaerobic metabolism. And then in response to injury, we still make the glial scar. This is still true. When there's a site of injury in the brain, the astrocytes are going to quarantine off that area, which I am not talking about right now. Scratch that from the record. This is the cleanup of neurotransmitters and why this matters. This is why glutamate doesn't operate by volume transmission. When you allow glutamate to escape the synapse, when you don't clean up the mess, bad things happen. And one of those bad things would be seizure, the other bad thing might be death. And here they're both happening, unfortunately. In these mice, they've knocked out glutamate transporters in astrocytes. No more of these little orange balls. These little orange balls are fairly important. They're not actually orange or balls, but they are important. The open circles are showing you wild-type mice. In other words, nothing different about them. You go get any mouse, it should look like this. This is how it should work. Glutamate transporter knockout mice are in the filled circles. Notice they don't quite grow the same. Their body weight never reaches that of a typical mouse. And they also don't live as long. So here's the survival curve. So you can see they're dying off fairly rapidly. And that's because of these seizures, the hyperexcitability of their neurons. It's great that neurons are active, but it's also nice when they're not inactive too. You don't want to have your neurons synchronously active all the time because you can have excitotoxicity, just like we talked about last semester. When your neurons die, eventually you die. When enough of them are gone, you're gone too. And so here's a mouse having a little seizure. Here they're recording seizure events in those mice with the glutamate transporter knocked out. In other words, no more glutamate transporter, seizure activity. Typically, you don't see seizures in mice. So these are really important. It is important to clean up that mess. Now we're on to the glial scar. So, in response to injury, imagine that I transitioned a little better. In response to injury, our astrocytes are going to create this barrier here that prevents the diffusion of inflammatory cytokines or foreign invaders, or toxins that they produce, or toxins from the blood. They prevent that from diffusing away from the site of injury by creating this glial scar so that the healthy tissue isn't affected by injury. The glia are there to take care of the neurons, and there are many ways that they do that. Do we have any questions? Oh, wait, I have one more. Before we move ahead. Then maybe you'll have questions, I don't know. Now, because astrocyte function is vital, not only for neurons to continue to work, but for the organism to live, as we've seen, it's important that they have a little support too. Parents need help from time to time. And so they're helping each other. The way that they do that is with gap junctions. They're back. Same gap junction we've talked about already. But rather than sharing electrical charge, this is more about sharing the ions that they pick up, or the neurotransmitter that they pick up, so that the astrocyte can buffer a greater quantity of ions and neurotransmitters. It's not just one acting in isolation. No, you create a network of astrocytes and the non-myelinating oligodendrocytes too. They join the party as well. But they all act together to help clean up. So if you have a local area of very high activity, you're spitting out a bunch of potassium, a bunch of glutamate, we need to clean this up. Just because it's here doesn't mean that this astrocyte can't help. It'll get taken up here and transported freely through gap junctions, so they can all act as a unit, increasing the buffering power of astrocytes. And it also allows them to share the metabolic substrates as well. So you got to feed the neuron. Neurons don't contact blood vessels, astrocytes do. You got to feed them. Well, what if I've given it all the pyruvate I can give? I'll borrow some from you, and you, and you, and you. And I can keep feeding these neurons. It takes a village. I don't know what that means, but it sounds good. Here's our village. Now, do we have any questions? I hope that's it. I don't want to pull a wing at the white coat ceremony here. Okay. Let's hear about our glia and just very basically what they do. Not too much, please. Harlan.
So you have the first ones are the ependymal cells. They make CSF.
Great.
Then you have the oligodendrocytes and Schwann cells. They kind of do the same thing in different places, but they protect axons and then increase transmission speed through
What is it?
myelination.
Yeah, there we go. Myelination.
Yep. Then you have microglia, which are the immune cells. So they protect it from any kind of bacteria or virus or anything.
Okay.
And then you have astrocytes, which are, like you said, are kind of like the mom, so they clean up after, bring the nutrients, kiss the boo-boos, make it to the star, take it from
I'm
the
gonna steal that one. Yeah, that's pretty good. Okay, you mentioned oligodendrocytes and Schwann cells, and you said they do kind of the same thing in different places?
Yes.
What do you mean by that?
So the oligodendrocytes are in the central nervous system, and they wrap multiple axons,
Okay.
so you can pack in tighter, whereas the Schwann cells are in the PNS, and they only wrap one axon at a time, and that's because it's in the PNS, so it's more likely to have injury, so it's more of a cushion.
Great. More of a cushion, exactly. Kind of fluffy myelin, less compact. Yeah. Okay. Why do we care about gap junctions in astrocytes? Daniel.
Because we need a more robust buffering action to help support the neurons. They're not directly near them.
There you go. Yeah. So they're sharing their cytoplasm. They're sharing their burden as well. Very good.
All stick together.
Yeah. Okay, let's talk about what astrocytes and microglia do in response to injury. Kate.
So astrocytes would come in to clean up after wrong way. Microglia would come in to clean up, and astrocytes would help make like a glial scar to confine it.
There we go. That's, I think, a pretty good division of labor there. Yeah. Any questions? All right, well, thank you for your attention. There'll be a post quiz posted now that we've had the lecture.
So we're.
Take that before Thursday. Please and thank you. Before lecture. No, you'll click, it's another Google form. Yeah. Anything else?
Is it like the best work from pre and post?
Yeah, exactly. So in case you bombed the quiz, no big deal. Pay attention, do better on that.